Hi, in this video we are going to see about my research work entitled Receptor and Mitochondria Targeted Photodynamic Therapy using Ligand-conjugated Bacteriochlorin Photosensitizers for Cancer. Let's see how it works. This is a representative image of a cell. We can see there are different types of receptors present in the cell membrane. However, our conjugate is selective to cancer cell receptors. Our conjugate consists of 4 essential components. 1. Payload. 2. Small molecule ligand. 3. Linker. 4. Cleavable linker. The payload contains the therapeutic cargo and the mitochondria targeting moiety. The small molecule ligand is highly selective to cancer cell-specific receptor. The linker is used to conjugate the payload and the ligand, and the cleavable linker can be cleaved once it enters the cancer cells by cancer-specific enzymes. Since the ligand is highly specific to cancer cell receptor, it targets the cancer cells while leaving the normal cells untouched. Then it leads to formation of receptor-ligand complex. This receptor-ligand complex then enters the cancer cells through endocytosis. Endocytosis is a process in which the endosomes engulf the receptor-ligand complex and deliver it to the cytoplasm. During endocytosis, the receptor-ligand complex gets cleaved inside the endosomes by endolytic enzymes. Eventually, the conjugate released inside the cytoplasm gets cleaved by cancer-specific enzymes such as GSH. Then the cationic mitochondria targeting moiety available in the payload directs the payload towards mitochondria. Once the payload reaches the mitochondria, a specific wavelength of light respective to the synthesized bacteriochlorin is delivered through a laser source, typically in the phototherapeutic window. As a result of photon absorption, the bacteriochlorin photosensitizer gets excited. The excited photosensitizer generates reactive oxygen species such as hydroxyl radical, hydrogen peroxide, superoxide anion, and more importantly singlet oxygen. All these species are toxic to cancer cells, which leads to death of cancer cells. As a result of photodynamic therapy, 3 things can happen. 1. Cell death through apoptosis, necrosis, and autophagy. Apoptosis and necrosis are the 2 major cell death pathways, while autophagy is a degradation process in which essential cell organelles are self-digested and removed. Simply, the cancer cells will die as a result of photodynamic therapy. 2. Activation of immune responses. Photodynamic therapy can enhance the immunity in the tumor microenvironment by inducing local inflammation and recruiting cytokines. It also promotes the infiltration of immune cells such as neutrophils, macrophages, dendritic cells, cytotoxic NK cells, and T cells, which results in enhanced immune responses against cancer cells. 3. Blockade of angiogenesis. Angiogenesis means formation of new blood vessels, which is a hallmark of tumor development. Angiogenesis is required for the development and survival of tumor cells. Photodynamic therapy can shut down angiogenesis, which may also lead to death of cancer cells. In summary, the ligand-conjugated bacteriochlorin photosensitizers administered to the cancer patients. And it reaches the bloodstream. Then it targets the cancer-specific receptors present in cancer cells. Once it enters the cancer cells, the payload gets cleaved and reaches the mitochondria. Then a specific wavelength of light is passed to excite the photosensitizer. It leads to generation of reactive oxygen species, which eventually results in the death of cancer cells. The overall objective of my project work is complete eradication of cancer.